Right, good morning everyone. Thank you so much for the invitation. Unfortunately, the PI of the DMAC wasn't able to be with us, so you get me as a consolation prize. So, this morning I want to, Lashley's quite nicely outlined the three projects that are part of our program. And so I want to just focus a bit on the data core, the data management and analysis core, and the NGS core, and how we are facilitating and supporting the data so that the three projects can be successful. So I'm here on behalf of the DMAC, and the project involves multiple organizations, as Lashley has already told us. The primary grant holder is the Institute of Human Virology in Nigeria, and we also are collaborating with the University of Maryland, CERI in Durban, and then the private partner is Across, and then the University of Stellenbosch is the manager of the data. So this morning I'm just going to give a quick overview of what the DMAC aims to do, where our data is coming from, look a little bit about our data management plan and how the data is stored. I'm going to give an example of the NGS data, to give an example how the analysis will occur, and then just talk a little bit about available computer infrastructure that we have, and touch on some of the ethical considerations in doing a project like this. So firstly, just to orientate us, the DMAC, where it fits into this whole project is we're a core that supports all of the projects as part of this initiative, but we also interact with the wider DSI through the LC, which is REDSSA, as well as the Open Data Science. And that is hosted by Nicola Mulder at UCT, and that project's called the Elawazi. So a lot of the analysis and interaction in the data occurs through that structure. So the main goal of the DMAC is to provide this platform where the projects can interact seamlessly. So the first aim is to develop this scalable platform that can coordinate all the data and the tools and the workflow components. The next aim we have is to come up with the data sets that can be used for the analysis for each of the three projects. But then we also, the NGS part of the core is to provide the sequencing needed for of COVID for the various projects. And then the last aim is to provide training and support across the projects in data analysis. And so where does the data come from? So as Lashley outlined, this project is done in conjunction with Nigeria. So it's data, matching data from both Nigeria and South Africa to better understand and use that data to prepare for the next epidemics. But we're accessing multiple sources from both private and public partners. And it's population scale data, so it's millions of data fields. And for the COVID data, we're looking at national household seroprevalence surveys, so a matching survey in Nigeria and South Africa. And then for the testing data, we're looking at, again, a national database. So in Nigeria, this comes from the Nigerian CDC, and from South Africa, it comes from our National Institute for Communicable Diseases. And then they also access some open source data from this government measurements data set. And the one aspect that we have on the South African side is to bring in the private sector. So we have a collaboration also with Discovery Health to look at data from Discovery Health and add it to the public sector data as well. For HIV, so in the third project, we want to look at the interaction between both HIV and COVID. So there's some aspects of HIV prevalence that we are interested in, but also linking measures of things like HIV viral loads. So the data sources will be through, again, national surveys and through the government systems. The project also involves the use of mobile data, so there's a project dedicated to mobility, and there's multiple sources where this information comes from. There's also data coming from the World Bank surveys, which is an open source data, and then the genomics is coming mainly from ASCID and CERI. And the ASCID is a lab in Nigeria, and all of the sequencing information that is produced is uploaded into GISAID, which is available open source. So how are we going to manage this data? So all of the data coming from the various sources will be managed by the Stellenbosch group, and depending on the type of data, it will either be come into the data center through APIs or through encrypted data uploads, depending on the source. And then the data will be tokenized to anonymize it, and it will go through a cleaning process trying to coordinate the data from both Nigeria and South Africa, to make it into a database that can be used then for the analysis. All of this data management will happen through the Terra system, which is it's an online platform where you can interact and you have a workspace, and this is managed by the Elewazi team, and then we can then share the data with our various partners. So just giving an example on how the next generation sequencing, how the analysis would work for that, we would have, so far we have about 42,000 SARS-CoV-2 genomes, and that's in GISAID, so it's already publicly available. That is then combined with some metadata from the patient, which includes things like the sampling date, location, perhaps gender and age, travel history, comorbidities, vaccination status, chronic infection status, and CT score. So it's never a name and never an ID number, so we can't link it to an individual. We also have access to global information through GISAID, and then of course you want to make sense of the variants, so you link it to the epidemiology, that's your cases, your deaths, excess deaths, your positivity rate, hospitalizations, your S gene target failure proportions, and your non-pharmaceutical interventions that are present at the time. So what do we do with all of this information? But we want to combine all the different elements to make sense of it so that it's useful. And so there'll be some data analytics, but some of the actions that can arise from combining all of this data is we could optimize sequencing. So some of the information that comes out, you can improve the way that the sequences come out. To detect new variants, which has been quite important in COVID. Also outbreaks, if you have a hospital outbreak, genomics is important there, but also just continuous genomic monitoring in the epidemic, but also to alert the rest of the world if we are seeing something that is unusual. So how does this all look? So the data looks like this, which is just code of the virus, and then there's some R scripts that very clever people write to make some sense out of the data, and it can produce things like these diagrams, so you can look at the trends in variants over time, you can look at transmission dynamics, how variants are coming into the country, where they're spreading, and just useful aspects of how you analyze the sequencing data. So where are we keeping all this data, and specifically the infrastructure available for the sequencing? So at CERI we have a very high throughput sequencing capacity. So we have 5 sequencers, including these 2 NovaSeq machines, which are the Rolls-Royce of sequencing, I'm told. But they are capable of doing very high throughput sequencing. So in one single run, for example, they can do 5,000 whole viral genomes and even 100 whole human genomes. So sequencing so many genomes, you can imagine it generates an enormous amount of data, terabytes of data. So to help with the processing of all this data, we have a Dragon Bio IT system that sits next to the sequencers, which allows the assembly of the genomes quite quickly compared to another server, which would take hours and hours. It cuts down the analysis time 100-fold, and the file size about 1,000-fold. We also have local storage for the sequences, and then processing happens on Mac computers. But the data is also stored off-site at the MRC, at their data center, and it's stored on these Linux clusters, and they have these solid-state storage facilities which are very secure, and then the data is then made available can be made available through the Terra network in a cloud part. So just to speak briefly about some of the ethical considerations. So the study has been approved by 3 ethics committees so far. So the University of Maryland, the Nigerian National Health Research Ethics Committee, and we've just received approval from the UKZN BREC, and we will now submit a reciprocal ethics now that we've received the approval from BREC. So the study uses anonymized data, so we don't have any personal identifiers in the study. So the data we receive from NICD has no name, no ID number. Any linkage to any perhaps viral load happens on their side before the data comes to us. So we've requested an informed consent waiver. There are some aspects of the project where they will be asking for a survey, and we have a specific informed consent specifically for that data. So when they launch that particular part of the project, we will have an informed consent for the data collected as part of that survey. So the data will be secured but through anonymization and this tokenization of the data to prevent unauthorized access. But we're also using this Terra platform that is used in many other fields, including the H3 Bionet Africa. And this system is accredited and authorized by Federal Information Security Management Act, but it goes through continual compliance. And then just lastly, the ensuring data privacy, we will comply with the laws in South Africa, which for us is the POPI Act, but also in Nigeria, which is the National Health Act, but all of the data is de-identified. And if there is any printed information, it will be secured in a locked file cabinet, but we don't anticipate we're going to have any printed information from this study. So just to acknowledge the funders that are funding the project, which is the NIH, and then we have many people working on this project based in Nigeria, in the US, and then in Zambia, which is ACROS, and then our 2 partners in South Africa is CEPRISA and Stellenbosch University, and then our partnerships with the government organizations and and other research organizations. So thank you so much.